So, as I said, today now we are going to see the supercoiling of DNA. Without much ado, let us have a look into the video. It's a very good three-dimensional video which will give you idea about three-dimensional arrangement of DNA. So,
in this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell.
Okay.
The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
So, class, was it helpful for you to understand about the structure of DNA and how it is packed into a model, that is solenoid model? So now we will move towards structure of RNA. How RNA is there and what is the difference between RNA and DNA, that we are going to see now. Okay. So the basic difference between RNA and DNA is the word D and R. Na is same as nucleic acid. RNA stands for ribose nucleic acid and DNA stands for deoxyribose nucleic acid. While I was showing you the structure of nucleic acids, that is the structure of pentose sugar, deoxyribose and ribose, I told you one oxygen is removed from ribose sugar to give you deoxyribose molecule. So this deoxyribose molecule is present in DNA and in RNA it is ribose. RNA is a polymer. So what are the other parts? In this case also there will be presence of nucleotide and nucleoside. In 1967, so in 1953, that was the year when structure of DNA was proposed by Watson and Crick. Although it took years for them to grab the Nobel Prize, but actually they proposed the structure of DNA earlier. So after that, the Carl Woese, who found out about the catalytic DNA, and discovery of catalytic DNA changed the idea, sorry, catalytic RNA changed the idea of structure of RNA and its function. Okay, so catalytic RNAs are known as ribozyme. What are ribozymes? As the name itself is suggesting, it's having two parts here. One is ribo, another is zyme, which is abbreviation of enzyme. So this type of RNA is having property of RNA as well as property of enzyme. This was a breakthrough discovery in the field of RNA technology and helped us to understand many things. Apart from that, RNA structure is based on, RNA is made up of, I would like to add here, ribose sugar plus nitrogenous bases. Now, one difference of RNA DNA is this, that deoxyribose ribose. Second difference lies here. In case of RNA, the nitrogenous bases present there are adenine, guanine, cytosine, and instead of thiamine, they have uracil. In RNA, never ever thiamine or T is present. So pairing is, in RNA, there is no pairing also because generally RNA is single stranded. Only in case of virus 5x174, the RNA can be double stranded too. So in single stranded RNA, how they are arranged? Basically same way, sugar will be there and two sugars are connected with each other via phosphate. And the same, that is 5' and 3' position. Okay, and at the first carbon there will be presence of nitrogenous bases. Rest of the structure is similar, the bonding type is similar, only difference is difference in the pentose sugar and the difference in the nitrogenous bases and difference in single stranded nature of the RNA. Is this clear to everyone? Class?
Yes, ma'am.
So we will move ahead. There are, this is showing the basic difference between RNA and DNA. It's pictorially depicted here nicely, very nicely. And this is the detail of the RNA structure. I told you. This is how nucleotides are formed by addition of phosphate, the ribose sugar and nitrogenous bases. This is how two ribose sugars are connected with each other. The link which is operational here is called phosphodiester linkage. It is same with the DNA molecule. This is how RNA is there. As I told you that RNA, as it is single stranded, there is no chances of double strand or pairing between nitrogenous bases. But if in case of any virus pairing is possible, then in that case, similar to DNA, in RNA also hydrogen bonding will be there. In that case, adenine will pair with two hydrogen bond to uracil and guanine will be pairing with the 3 hydrogen bond with cytosine. So this is the structure of double-stranded RNA. Actually, let me tell you one fact. Although RNA is single-stranded beta, but the problem is many a times it is observed that single-stranded at a stage in the form of a straight wire, it cannot exist. It needs to pack itself. So here in this image, you can see that various types of loops possible in case of RNA. That means consider a structure which is long like this, having Now, consider a situation if you can look here, these two parts are complementary, isn't it? Is these two parts complementary or not, beta?
Ma'am, here it is a GCC.
Oh, it will be CGG.
Oh.
Sorry. So now is these two parts complementary?
Yes, ma'am.
So what happens in when natural condition, this RNA will be there, it will form a loop between these two parts and form a structure like this. That means AUG, GC, C. Here the structures like U will be remaining, and here at this part it will internally fold like this. Okay, these types of loops are very common, although it doesn't have a single double-stranded structure and standard one like DNA, but wherever and whenever it is having complementary part in between, the RNA can depending on complementary part, it can obtain various structure. So the structures described here are different types of such structure where see loops are formed or there is formation of a bulge. Why bulge? Because this part, why bulge? Because this part is complementary, this part is complementary, but this part is not. So this part is staying away from each other, so it's a bulge, whereas the part which is complementary is forming a structure like this. Here you can say 4 RNA strands are there. 4 of them is having alternate complementary structure. For example, this one is having this part complementary to 4 and this part complementary to 2. Understood? Is this clear to everyone?
Yes, ma'am.
Similarly, 2 is having this part complementary to 1 and this part complementary to 3. This way, multi-branched pairing between different part of RNA is possible to create junction-like structure. This is known as X junction or holiday junction. Similarly, in this type of RNA, what happened is this is one strand, some other RNA piece is there which is having loop-like structure. So this is hairpin structure, multi-branch junction, internal loop, bulge loop. These are the cases which may be present even in single-stranded RNA depending on the complementary sequences. Now we will come to the part that is types of RNA. The way DNA can exist in different forms. Those were forms of DNA. Same DNA can exist in the form of A DNA, B DNA or Z DNA. We discussed about it yesterday. RNA can also exist in the form of messenger RNA, transfer RNA and ribosomal RNA. But let me tell you the way DNA can exchange the form. For example, a piece of DNA which is depending in the situation when there is dehydration existing B form, when is water it is existing in A form. Same DNA can obtain form. It's not the case in case of RNA. In case of RNA, messenger RNA are specialized RNA. They have a specific type of structure and function. Transfer RNA, another type of RNA. It's never occurring that messenger RNA is taking shape of transfer RNA and vice versa. Okay. So these are the three main types of RNA. Apart from that, there can be small nuclear RNA, sRNA. They are also called SNRPs or ribozyme. Apart from that, miRNA is there, siRNA is there, and hnRNA is there. It will be a bit difficult for you people to remember about each of them. Now I'm going to describe the structure, but in this chapter itself, we are going to read separately about messenger RNA, transfer RNA, and ribosomal RNA. At that time, it will be easier for you to understand the difference and to understand their function. So let's talk about messenger RNA first. That day I was telling you that DNA is the genetic material. It is responsible for carrying information from one generation to another generation. Isn't it? Yes or no?
Yes, ma'am.
So, if DNA is present inside nucleus and ribosome is the center for creation of protein, is it possible for the DNA to come out of nucleus and pass on the ribosome the information by using which it can produce protein? Do you think it will be a nice idea? अच्छा आइडिया रहेगा ये डीएनए के लिए हर बार बाहर आना? And remember the situation, there is not only one ribosome. There will be thousands of the ribosomes. ये सभी क्या करेंगे? They will be waiting for the instruction of DNA to create the protein. Because I told you the sequence of DNA will decide the sequence of amino acids of protein. So do you think it will be a good idea for DNA to come out? What do you think?
No, ma'am.
So how to solve this problem? The way if you wish to carry one movie or one important document or one video from your friend's system to your system, we don't uproot the whole system. We copy it into some other means and share. We copy it either into drive or into this one, what I can say, into pen drive. Same way, DNA, the part which is to be sent to ribosome is copied in a piece of RNA. That's why this RNA is called messenger RNA. And then messenger RNA will carry the message to the ribosome. So generally, this is the structure of messenger RNA. That 5' terminal is capped. That means there is a protecting part added to it, which is 7-methyl guanosine triphosphate. That means the last guanine of the RNA is having methyl group and 3 phosphate group. So that is how it is capped. Apart from this, why this cap? Because it is helpful for the RNA to recognize, for the ribosome or other part to recognize the messenger RNA. Okay, and RNA is having a polymer of AD. That means if considered, this is the 5' end, it will have a cap over here, and at the end of this messenger RNA, there will be a long tail which is having A only. That's called poly A tail. अब इतनी लंबी A के tail का क्या काम? Is it the message? No. When the RNA is coming outside nucleus, there are chances कि कोई इसके पीछे पड़ जाए और इसका थोड़ा सा part digest कर जाए. Okay, so to prevent it from the activity of 3' exonucleases, this mockery or tail is added. जैसे छिपकली का होता है, मुसीबत देखते ही वो उसको छोड़ भी दे तो उसको कोई फर्क नहीं पड़ता है. यहाँ पे अगर समझो, if it is a long AAA, if an exonuclease is chewing a few number of A's, the original information is not hampered. That's why it is having a poly A tail. हम लेटर भेजते हैं तो एनवेलप में भर के भेजते हैं कि नहीं भेजते हैं? Yes or no? Why we do not
Yes, ma'am.
send it without cover? Because cover is protecting it from getting damaged. Same is there for the poly A tail responsibility. So this is how messenger RNA looks. Let's have a look into the other types of RNA. When actually the DNA part is getting copied into messenger RNA, this is how the process takes place. See, this is the RNA, DNA from which this is the RNA produced with a start code and stop code. HNRNA is a precursor of messenger RNA. When this is existing only in case of eukaryotes. HNRNA is a bit bigger than the messenger RNA. If you can remember, that day I told you that mouse and human being is lot many similar, isn't it? If you can remember, I told you about junk DNA. What is junk DNA? Or non-coding DNA? What's its significance? Can you tell me? The DNA content we have in our body, out of that DNA content, much part is not coding at all for any protein. It's just there, they are not coding for any protein. So if I consider a part of DNA present in eukaryotes, some part of it is coding for protein and some part of it is non-coded coding for protein. So the part which is having meaningful implication is called exon, represented by E, whereas the part which is not having meaningful part is known as intron. So what happens is here, they remove the not useful part and create the messenger RNA. This is prominent in case of eukaryotes. So each RNA has both the useful and non-useful part of RNA, and messenger RNA has only the useful part of RNA. That is what the importance of messenger RNA and HNRNA. Is this part clear to all of you?
Yes, ma'am.
Now transfer RNA.
मतलब
Yes, beta.
मतलब एचएनआरएनए में से जो इंट्रॉन वाला पार्ट है वो निकाल देंगे तो मैसेंजर आरएनए बन जाएगा.
बन जाएगा. Only messenger RNA have only the useful part. Okay.
Okay,
so that this process is called splicing. Again, in this chapter only we will learn about it. Just giving you a brief idea because we are talking about types of RNA. Next is transfer RNA. The way trucks load the goods and send it to one place, role of transfer RNA is same. The 20 amino acids are carried by different sequence of transfer RNA. What they do is they carry the amino acids and supply it to the ribosome so that amino acids can be added as per the codes present in the DNA. I told you that in DNA the codes are in the form of triplet. जैसे कि AUG आया, that means the start codon, which represent for methionine. तो जैसे ही AUG आएगा, एक ट्रांसफर RNA जो अपने पास एक methionine को carry कर रहा है, that methionine containing RNA will be placed when AUG code is there. Next code, next amino acid. Next code, next amino acid. To match with the code of gene or triplet code, As per the codes present there, transfer RNA is going to supply the amino acid. So basically it's goods carrying truck kind of. This is the structure of transfer RNA. Obviously, you can see these loops are here. These loops are here because these are the parts where complementary sequence is there. Okay, this is the part, this beta part is mentioned here. Actually the amino acid is binding here, and here the sequence is there, that is AUG. तो समझो अगर if the messenger RNA is having A, U, G, this one will have U, A, C. So using these complementary relationship, जैसे ही AUG आएगा वो यहाँ पे मिथियोनिन को कैरी करेगा. It will carry methionine. So all of these arms are having their names. It's known as anticodon arm. This arm is known as anticodon arm. Okay. Then acceptor arm. Then there will be T5C arm. There will be DHU arm. And this arm specifically is optional. It may be present, it may not be present. It's not always present in all tRNA. Okay. So this is how the structure of tRNA looks. This model is also known as clover leaf model because it looks like a leaf of clover. Clover की जो leaf होती है उसमें से चार, it has 4 leaflets. So it somehow look like that. That's why it's also known as clover leaf model of transfer RNA. Is this part clear to all of you?
Ma'am.
Yes, Dish.
Ma'am, so the starting code is AUG.
Yes.
मैम सो हमको कैसे पता कि उसके बाद वाली कोड कौन सी होगी?
मुझे नहीं पता चलना है बिल्कुल. See, we will talk about it in detail when we study protein synthesis, but just to give you a brief idea, ये एक रिलेशनशिप है. जितनी भी कोड्स होते हैं, जैसे समझो अगर समझो कोई कोड है यू A C. So there will be a transfer RNA which is having complementary to this AUG present, which will carry a specific amino acid that is to be there. It's a code. जैसे कोडिंग हम बोलते हैं ना, कोडिंग लैंग्वेज में 0 का क्या, 011 का क्या मीनिंग होता है, 101 का क्या मीनिंग होता है, कंप्यूटर कोडिंग लैंग्वेज में सिमिलर टाइप ऑफ कोड इज देयर. I don't have to do it anything, but it was an Indian scientist working with other people. He like discovered genetic code that which amino acid is coded by which triplet code. His name is Hargobind Khorana. Okay, so that code I don't do it any consciously. It is discovered. It was discovered in fact, and specific transfer RNA and come and bind with the specific code present here, and it's carrying the specific amino acid over here. The exact process, these we will learn in this chapter only. There is a part called protein synthesis.
Okay, ma'am.
Any more doubt, Dish?
No, ma'am.
Great. So we will move ahead. This is how it looks. This is the tertiary structure. That means the three-dimensional structure. This is how it looks actually, the global leaf model. Ribosomal RNA or rRNA, as its name itself is suggesting, ribosomal RNAs are associated with ribosome. These ribosomal RNAs will help in the binding of mRNA with the ribosome, and it is forming the 60% of the constitution of the ribosomes. So they are various types, and some of their sequence is very much conserved. By 16S or 18S rRNA analysis, we can predict the ancestral lineage of any organism. जैसे कि evolution में हम किससे related थे? There is a comment that we were related to this, we were related to that. Evolutionary people, like evolutionists, comment on that. That is done on the basis of analysis of these ribosomal RNA. Small RNA molecules can be small nuclear RNA. They help in RNA splicing. That is, I told you that from hnRNA to messenger RNA formation. That is removal of the unnecessary part. That part is done by small RNA molecules. Apart from that, there are micro RNAs, and they regulate the gene expression. That means I am saying that, okay, I have this character in DNA, but it should be expressed in a character. For example, if in DNA the signal is there that the complexion of a person should be dark, so for that, that should be translated to the language of the phenotype. That is, that character should be expressed. So this miRNA is there for expression of gene, and snRNA like help in the modification of ribosomal RNA. So this is the difference between RNA and DNA. I want you people to go through each point and let me know if you have any doubt. Any doubts, anyone? Can I change so that we can look into other points? Class?
Yes, ma'am.
So let us, I hope the difference between RNA and DNA is clear to all of you. Or is there any doubt in any point? It's a long difference. We jotted down all the characteristics, but at least 3 to 4 you should remember. And I mentioned about them already. So with this, we are done with the structure of DNA and RNA. Anyone, any doubt?
A beautiful double helix structure. Sometimes it feels like DNA gets all the credit. Yes, the structure of DNA is amazing. It has a beautiful double helix structure, a twisted ladder, it has its own emoji. DNA stores genetic information and codes for your traits. However, sometimes what gets left out is how important RNA is. Without RNA, you actually couldn't get that genetic message out to your cells so that they can start producing proteins. We talk about this in protein synthesis. RNA is a very important biomolecule, just as important as DNA. In fact, RNA is even hypothesized as coming first, before DNA, in the RNA world hypothesis, something we need to make a separate video about. So let's compare and contrast DNA with RNA. First, you will find DNA and RNA in all living organisms. In eukaryotic cells, DNA tends to be found in the nucleus, while you can find RNA both inside and outside of the nucleus. Prokaryotic cells don't have a nucleus. Both DNA and RNA are nucleic acids, which are a type of biomolecule. Nucleic acids have a monomer, which, if you remember from our biomolecules video, a monomer is a building block. The monomer for nucleic acids is a nucleotide, so both DNA and RNA have nucleotides. The nucleotides of both RNA and DNA have 3 parts: a phosphate, sugar, and a base. Let's draw 6 nucleotides of DNA and 6 nucleotides of RNA. DNA is generally double-stranded, and if focusing on these 2 strands of nucleotides here, you can see they run anti-parallel to each other. RNA is generally single-stranded, so you are just seeing one strand here. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. This makes sense because DNA stands for deoxyribonucleic acid. That's helpful to know because the deoxyribose is a sugar, and nucleic acid is that type of biomolecule it is. RNA stands for ribonucleic acid, as its sugar is ribose. The bases in DNA are adenine, thymine, guanine, and cytosine. It helps to remember the popular mnemonic apples in the tree. That helps you remember that A goes with T, and car in the garage, so C goes with G, to understand how DNA bases pair. The bases in RNA are adenine, uracil, guanine, and cytosine. Notice the different one? It's uracil. So you have to change that popular mnemonic device here. Instead of apples in the tree, maybe the apples are under. Car in the garage still works though. We mentioned earlier that DNA codes for your traits, but it couldn't do that without RNA's help. In our protein synthesis video, we talk about 3 different types of RNA, and their very important roles. mRNA, which stands for messenger RNA, mRNA's job is to carry a message based off of the DNA. In eukaryotic cells, DNA generally stays in the nucleus, but mRNA has the ability to leave the nucleus to take the message to a ribosome. Ribosomes make protein, and RNA is actually a major component of ribosomes. This type of RNA is called rRNA, which stands for ribosomal RNA. Finally, we discussed transfer RNA, or abbreviated tRNA. Its job is to transfer amino acids to match the correct mRNA codon. Codon charts using mRNA codons have been developed so that you can actually see which amino acid is brought for each mRNA codon. When those amino acids are joined together, they make a polypeptide chain. Proteins are made of one or more of these polypeptide chains, and proteins have tons of different roles. But we don't want to spoil it. Check out more in our protein synthesis video. Before we go, let's try a little 3-question quiz, shall we? Just pause it after the question so that you have time to think about it. Question 1. If I have 8 DNA nucleotides, how many DNA bases do I have? And also, how many base pairs? The answer: each nucleotide, regardless of whether it's a DNA or RNA nucleotide, has a phosphate, sugar, and a base. So 8 DNA nucleotides would have 8 bases. DNA bases pair like this, and that's 4 DNA base pairs. Question 2. If one strand of DNA has these bases shown here, ATTGAC. Can you complete what the complementary DNA bases would be for the other DNA strand?
So I wish to ask this question to someone specific. Siddhant, can you answer this question? Siddhant, are you there? Any person who can speak and answer other than these Shiesha and Jia? Because these people speak at least in the class. Kashish, Kausha, Shama, these I know, but you will be able to say. I need to look into the others also, no? Anyone else? If in one strand the sequence is ATTGAC, in other strand what it is? Okay, Dish, you speak better.
Ma'am, the strands would be TAACTG.
Okay. So others I hope understood why, because in other strand A is complementary to T, vice versa, and G is complementary to C. So that's all from my side. Now we will.
The answer. So remember those base pairing rules for DNA and the popular mnemonic. If placing the bases in this image, reading in the direction shown on this image, they'd be TAACTG. Question 3. In a process known as transcription, a complementary RNA strand called messenger RNA has to complement the DNA. More about that in our protein synthesis video. So if I still had the original portion of DNA here, what would be the complementary RNA bases?
Second question of mine. If this is the strand of DNA, what will be the complementary RNA base? Class? Yes, Yesha. I can see three
Ma'am,
participants. Wait. Yes, speak, Yesha. You can proceed, Yesha.
yes, ma'am. Ma'am, am I audible?
Yes.
Ma'am, the strands would be like U, A, A, C, U, G.
Very good. So for RNA, it is always U, not T. That's what we need to remember.
The complementary RNA bases. The
It's
answer. So remember, it's asking for
correct,
RNA.
Yesha.
No thymine here, it's uracil instead. Apples under helps you remember A with U. Car in the garage helps you remember C with G. So in this portion, reading in the direction shown on this image, we'd have these RNA bases. Notice the answer is similar to the last answer, but there are uracil bases here instead of thymine. One last thing. Our models here, they're unable to show the beautiful 3D shape, the exact numbers of bases per turn, the chirality, but we've left you some links to explore because the goal of our videos is always to introduce the topic so that you can discover all the fascinating details and exceptions. Check out our further reading description to keep that curiosity going. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So I hope it was a funny way. They shared about the concept of car in the garage, and by that means they said that the C is pairing with the G, and they say that the apple is under the tree. So by which they said that the A is pairing with U and C. So I hope there should not be any doubt, but if you have any, then please feel free to ask. Let us learn more about the process of DNA replication from this part. Okay, so DNA replication means what? DNA copying. DNA replication, the another word is it copying. So why we need to copy DNA? What do you think should be the necessity of to copy DNA? Class?
Ma'am, so it can be passed to other generations.
Very good. We studied that two types of cell divisions are there in mitosis or in meiosis. Cell is divided into two cells, or in meiosis cell is divided into 4 cells. Now in either of the cases, the genetic material of a single cell will not be enough to be distributed into several cells. For example, if consider this cell is having 11 chromosome numbers or 10 chromosomes, it can distribute 5 chromosomes only to the next types of cells in mitosis. Similarly, if this cell is having 10 number of chromosomes, it can give each chromosome 2.5, 2.5, 2.5, 2.5 chromosomes. But does it occur? No. We see that in case of meiosis, although the chromosome number is reduced, but each of the daughter cells will have chromosome number 5, 5, 5, 5, not 2.5. And in case of mitosis also, the chromosome numbers will be not 5, 5, it will be 10, 10. So what is the sum total of 10, 10? It is 20. Here if I total 5 of these, it is 20. But initially I had only 10 DNA, 10 chromosomes in case of both the cells. So how, from where this extra 10 set of DNA is coming? That is, chromosome is coming. We need to understand, know that. So for obtaining that extra 10 chromosomes, we need to copy the DNA, because DNA is the basic of chromosome. Other things present in the chromosome is accessory, but the main thing of chromosome is DNA. Now, if DNA copying is stopped in a cell, then that organism will die. There is no chance of survival of that organism, because even if it is a unicellular organism, or in case of a multicellular organism, replication of DNA is required. In case of unicellular organism, the production of new types of cell is nothing but the reproduction. When in amoeba, by binary fission from one cell, there is production of two cells. During that procedure, DNA replication is must, otherwise both the cells will die. Similarly, in multicellular organism, if we have some part of our body is damaged or some part of our body require repairing, or we need to produce gamete for next, like to give next generation, we need to have DNA replication. Otherwise, this process will not be there. So this is the significance of DNA replication. So if this question is twisted and asked to you that what is the significance of DNA replication, you should be able to answer it. Now the next question is, when DNA replication takes place in a cell, what is the time when DNA replication takes place? So DNA has to be copied before the cell divides. So during mitosis or meiosis, the chromosomes is divided into different cells, and cell is having either half of the DNA content or 1/4 of the DNA content. Now, before this cell can go for the next set of division, cell require a replication of DNA. From this hint, can anyone tell me at what time the DNA replication takes place? You must have studied this in grade 11, beta. Anyone? Class? Which stage of DNA replication takes place?
S phase.
Yes, it is the est phase of interphase. That is synthesis phase of interphase when the DNA replication takes place. So that's why it's called preparatory phase. Between two mitoses or between two meiosis, the S phase or interphase is required. So at S phase, during interphase of the cell cycle, in nucleus of eukaryotes, this is how the cell cycle, this is the S phase. The DNA replication takes place. Now you have a question कि मैम अगर वो प्रोकैरियोट्स हुआ उसमें तो माइटोसिस मियोसिस नहीं होता है, they don't require DNA replication? No, they also require DNA replication, and in their case the DNA replication is a lot more simpler because they have less amount of DNA content and less complexity also. They don't have covering around their genetic material, so their process is comparatively very simple one. So today we will not start with the replication procedure. We will just take an introduction of the replication procedure here. So consider in DNA replication, it starts always from the point which is called origin of replication. It is denoted as ORI in a DNA structure. Now, in case of prokaryotes, as I told you, their DNA is comparatively smaller in size. In case of prokaryotes, one origin of replication is there. In case of eukaryotes, the DNA is so long that if replication process start at one position and we need to wait the process to be proceeded and covering up the whole genetic material, it will take years for the DNA replication. So in eukaryotes, many origin of replications are there. जैसे जो लोग साड़ी में डिजाइन बनाते हैं, you can see at many places it is observed that they spread the sari and 2-3 people are working in the same sari. So some person is doing for the first half, someone is doing for the next half, next half, next half. This is the way they work simultaneously for a small part, and when the whole work is done, you can see the complete preparation of the sari. Same is the place. Now the thing is that you need to replicate DNA. What you need to do? You need to copy DNA. So for copying DNA, consider if it is a double-stranded structure like this, or in prokaryotes it is having a circular structure like this. If you want to start the process of replication and you want to add newly synthesized DNA like this, consider this pink is the newly synthesized DNA. First thing you need to do is you need to separate the two strands of DNA. So before DNA replication start, we learn about that. We need to know that how separation of DNA is done. Is there the process of DNA replication continuous or semi-continuous? Is the process of DNA replication conservative or non-conservative? These answers. What are conservative replication? What are non-conservative replication? What do we mean by continuous, discontinuous? We will find this answer in our tomorrow's lecture, and we will talk about different experiments conducted by several scientists like Michelson and Stahl and Okazaki, and we will understand that how individual experiment performed by different scientists clarified the picture of DNA replication, and now we know the whole process of DNA replication in a form of a flow chart. So up to this introduction part, anyone any doubt?